Hello and welcome, dear students. Today's topic is coding and non-coding regions of DNA. The main objectives of today's lecture are to know about the coding regions of DNA, to know about the non-coding regions of DNA, and to know about the silencers, enhancers, and insulators. Genome is all genetic information of an organism. It consists of nucleotide sequences of DNA or RNA in RNA viruses. The nuclear genome includes protein coding genes and non-coding genes, the other functional regions of the genome. Alga and plants contain chloroplasts with a chloroplast genome, and almost all eukaryotes have mitochondria and a mitochondrial genome. The study of the genome is called genomics. The vast majority of the human genome is comprised of non-coding DNA, that is, genes only account for 1% of the total sequences which are coding. Non-coding DNA does not provide instructions for making the proteins. Historically, non-coding regions like satellite DNA, telomeres, introns, non-coding RNA genes, and gene regulatory sequences were referred to as junk DNA. But these non-coding regions are now recognized to serve other important functions within the cell. Therefore, the so-called junk DNA ends up playing a significant job in controlling genes, their regulation, and protein actions. Most purported junk DNA probably will not be junk anymore, and these puzzling components of the genome may have some important biological functions regardless of whether they do not undergo protein expression. In 1978, Walter Gilbert published Why Genes in Pieces, which first began to explore the idea that the gene is a mosaic, that each full nucleic acid strand is not coded continuously but is interrupted by silent non-coding regions. This was the first indication that there needed to be a distinction between the parts of the genome that code for proteins, now called coding regions, and those that do not code, which are known as non-coding regions. Coding DNA has genes that encode for protein. The coding region has the nucleotide sequence that codes for the proteins. They can transcribe, translate, and produce new proteins. These proteins have structural, functional, as well as regulatory importance in the cell. The percentage of coding DNA is less than the non-coding DNA. In DNA, the coding region is flanked by a promoter sequence on the 5' end of the template strand and the termination sequence on the 3' end. During transcription, the RNA polymerase or RNAP binds to the promoter sequence and moves along the template strand to the coding region. This RNA polymerase then adds RNA nucleotides complementary to the coding region in order to form the mRNA, substituting uracil in place of thymine. This continues until the RNA polymerase reaches the termination sequence. After transcription and maturation, the mature mRNA formed encompasses multiple parts important for its eventual translation into a protein. The coding region in an mRNA is flanked by the 5' untranslated region, that is 5' UTR, and 3' untranslated region, that is 3' UTR. The 5' cap and poly A tail, during translation, the ribosome facilitates the attachment of the tRNA to the coding region, that is 3 nucleotides at a time. The tRNA or transfers their associated amino acids to the growing polypeptide chain, eventually forming the protein defined in the initial DNA coding region. The protein encoded sequences of coding DNA are transcribed to mRNA. The mRNA is translated into amino acid sequences. These sequences make the proteins. The coding region of DNA contains genes that provide the instructions for protein synthesis. Genes are composed of regions that go on to form codons and hence are considered protein coding, and regions that are only there for regulatory reasons, meaning they do not code for proteins. Coding regions are referred to as exons, whereas the non-coding regions are known as introns. Protein coding regions in DNA are known to have a higher GC concentration than the rest of the DNA molecule. Within the gene, there are regions that form start and stop codon. The start codon is usually AUG, that is methionine, which signals the beginning of the translation process by coding for the amino acid methionine. Stop codons, on the other hand, signal the end of the translation process and the completion of protein synthesis through use of mRNA strand. Stop codons are either UAA, UGA, or UAG, Ocher, Amber, or Opal. Dear students, let's now discuss the regulation of coding regions. The coding region can be modified in order to regulate gene expression. Alkylation is one form of regulation of the coding region. The gene that would have been transcribed can be silenced by targeting a specific sequence. The bases in this sequence would be blocked using alkyl groups, which create the silencing effect. While the regulation of gene expression manages the abundance of RNA or protein made in a cell, the regulation of these mechanisms can be controlled by a regulatory sequence found before the open reading frame begins in a strand of DNA. The regulatory sequence will then determine the location and the time that expression will occur for a protein coding region. RNA splicing ultimately determines what part of the sequence becomes translated and expressed, and this process involves cutting out introns and putting together exons. Where the RNA spliceosome cuts, however, is guided by the recognition of splice sites, in particular the 5' splicing site, which is one of the substrates for the first step in splicing. The coding regions are within the exons, which become covalently joined together to form the mature messenger RNA. Let's now discuss the mutations in the coding regions. Mutations in the coding region can have very diverse effects on the phenotype of the organism. While some mutations in this region of DNA or RNA can result in advantageous change, others can be harmful and sometimes even lethal to an organism's survival. In contrast, changes in the coding region may not always result in detectable change in the phenotype. There are various forms of mutations that can occur in coding regions. One form is silent mutations, in which a change in nucleotide does not result in any change in amino acid after transcription and translation. There also exist nonsense mutations where base alterations in the coding region code for a premature stop codon, producing a shorter final protein. Point mutations or single base pair changes in the coding region that code for different amino acids during translation process are called missense mutations. Other types of mutations include frame shift mutations such as insertions or deletions. Dear students, Let's now discuss the non-coding DNA. The vast majority of the human genome is comprised of non-coding DNA, that is, genes only account for 1.5% of the total sequence. While it was previously thought to have no function, newer information is beginning to shed light on the many functions of this mass of DNA. It's involved in the cutting and splicing of large amounts of DNA, also involved in transposon reassembly, genome rearrangements, and the production of small RNAs, some of which may serve as source for new exons. It's also possible that non-coding DNA was used as a source of new genes needed for the adaptation or for functioning during the evolution. Non-coding DNA does not encode for proteins. Some non-coding DNA undergoes transcription to form non-coding RNA, may serve as a source for new exons. Non-coding DNA does not provide instructions for making proteins. While it was previously thought to have no function, newer information is beginning to shed light on many functions of this mass of DNA. The non-coding DNA include rRNA, tRNA, etc. Also, they regulate the protein coding sequences. The quantity of non-coding DNA greatly varies among species. Only about 1% of the DNA made up of protein coding genes. The other 99% is non-coding. Scientists once thought non-coding DNA was junk with no known purpose. However, it is becoming clear that at least some of it is integral to the function of cells, particularly the control of gene activity. For example, non-coding DNA contains sequences that act as regulatory elements, determining when and where genes are turned on and turned off. Such elements provide sites for specialized proteins called transcription factors to attach and either activate or repress the process by which the information from genes is turned into proteins, that is the transcription. Non-coding DNA contains many types of regulatory elements. Promoters provide binding sites for the protein machinery that carries out transcription. Promoters are typically found just ahead of the gene on the DNA strand. The RNA transcript may encode a protein, that is the mRNA, or can have a function in and of itself, such as tRNA or RNA, that is the transfer RNA or ribosomal RNA, respectively. Promoters are located near the transcription start sites of genes. Upstream on the DNA, that is towards the 5' region of the sense strand. Promoters can be about 100 to 1000 base pairs long, the sequence of which is highly dependent on the gene and the product of transcription. Type or class of RNA polymerase recruited to the site and species of organism. Promoters control gene expression in bacteria and eukaryotes. RNA polymerase must attach to DNA near a gene for transcription to occur. So promoter DNA sequences provide an enzyme binding site also. The 10 sequence is TAAT to 35 sequences are conserved on average but not in most promoters. Regulatory elements are sites that control the transcription of a nearby gene. They are almost always sequences where transcription factors bind to DNA, and these transcription factors can either activate transcription or repress transcription. Regulatory elements were discovered in the 1960s and their general characteristics were worked out in the 1970s by studying specific transcription factors in bacteria and bacteriophage. Promoters and regulatory sequences represent an abundant class of non-coding DNA, but they mostly consist of a collection of relatively short sequences, so they don't take up a very large fraction of the genome. The exact amount of regulatory DNA in mammalian genome is unclear because it is difficult to distinguish between spurious transcription factor binding sites and those that are functional. The binding characteristics of typical DNA binding proteins were characterized in the 1970s, and the biochemical properties of transcription factors predict that in cells with large genomes, the majority of binding sites will be fortuitous and not biologically functional. Enhancers provide binding sites for proteins that help activate transcription. Enhancers can be found on the DNA strand before or after the gene they control, sometimes far away from the actual site. Silencers provide binding sites for proteins that repress transcription. Like enhancers, silencers can be found before or after the gene they control and can be sometimes distance away on the DNA strand. Insulators provide binding sites for proteins that control transcription in a number of ways. Some prevent enhancers from aiding in transcription. Others prevent structural change in the DNA that repress gene activity. Some insulators can function as both an enhancer, blocker, and a barrier. Dear students, let's now discuss the untranslated regions. The standard biochemistry and molecular biology textbooks describe non-coding nucleotides in mRNA located between the 5' end of the gene and the translation initiation codon. These regions are called 5' untranslated regions or 5' UTRs. Similar regions called 3' untranslated regions or 3' UTRs are found at the end of the gene. The 5' UTRs and 3' UTRs are very short in bacteria, but there can be several hundred nucleotides in length in the eukaryotes. They contain short elements that control the initiation of translation, that is 5' UTRs, and transcription termination, that is 3' UTRs, as well as regulatory elements that may control mRNA stability, processing, and targeting to different regions of the cell. Other regions of non-coding DNA provide instructions for the formation of certain kinds of RNA molecules. RNA is a chemical cousin of DNA. Examples of specialized RNA molecules produced from non-coding DNA include transfer RNA and ribosomal RNA, which help assemble protein building blocks into a chain that forms a protein, microRNAs, which are short lengths of RNA that block the process of protein production, and long non-coding RNAs, which are longer lengths of RNA that have diverse roles in regulating gene activity. Dear students, let's now discuss the microRNAs. MicroRNAs are small non-coding RNAs with an average 22 nucleotides in length. Most miRNAs are transcribed from DNA sequences into primary miRNAs and processed into precursor miRNAs and then finally mature into miRNAs. In most cases, miRNAs interact with the 3' UTRs of target mRNAs to suppress expression. However, interaction of miRNAs with other regions, including the 5' UTR coding sequences and the gene promoters, have also been reported. Furthermore, microRNAs have been shown to activate gene expression under certain conditions. Recent studies have suggested that miRNAs are shuttled between different subcellular compartments to control the rate of translation and even transcription. miRNAs are critical for normal animal development and are involved in a variety of biological processes. Aberrant expression of these miRNAs is associated with many human diseases. In addition, miRNAs are secreted into extracellular fluids. So extracellular miRNAs have been widely reported as potential biomarkers for a variety of diseases, and they also serve as signaling molecules to mediate cell-cell communication. Dear students, let's now discuss telomeres and non-coding DNA. Some structural elements of chromosomes are also part of non-coding DNA. For example, repeated non-coding DNA sequences at the ends of chromosomes form telomeres. Telomeres protect the ends of the chromosome from being degraded during the copying of genetic material. Repetitive non-coding DNA sequences also form satellite DNA, which is a part of other structural elements. Satellite DNA is the basis of the centromere, which is the constriction point of the X-shaped chromosome pair. Satellite DNA also forms heterochromatin, which is densely packed DNA that is important for controlling gene activity and maintaining the structure of the entire chromosome. Dear students, let's now discuss introns as non-coding regions. An intron is any nucleotide sequence in a DNA molecule or within a gene that is removed by RNA processing during the production of the final RNA product. The word intron is derived from the term intragenic region, that is a region inside a gene. The term intron refers to both the DNA sequence within a gene and the corresponding RNA sequence in RNA transcripts. Introns are found in the genes of most organisms and many viruses, and that can be located in both protein coding genes and genes that are functional as RNA, that is non-coding genes. Dear students, let's now discuss their classification. Splicing of all intron containing RNA molecule is superficially similar. However, different types of introns were identified through the examination of intron structure by DNA sequence analysis, together with genetic and biochemical analysis of RNA splicing reactions. At least 4 distinct classes of introns have been identified till date. Introns in nuclear protein coding genes that are removed by spliceosomes. They are called spliceosome introns. Introns in nuclear and archaeal transfer RNA genes that are removed by proteins, they are called tRNA introns. Self-splicing group 1 introns that are removed by RNA catalysis. Self-splicing group 2 introns that are removed by RNA catalysis. So group 3 introns are proposed to be a 5th family, but little is known about the biochemical apparatus that mediates their splicing. They appear to be related to group 2 introns and possibly to spliceosomal introns. The spliceosomal introns are characterized by specific intron sequences located at the boundaries between introns and exons. These sequences are recognized by spliceosomal RNA molecules when the splicing reactions are initiated. In addition, they contain a branch point, a particular nucleotide sequence near the 3' end of the intron that becomes covalently linked to the 5' end of the intron during the splicing process, generating a branched intron. Apart from these 3 short conserved elements, nuclear pre-mRNA intron sequences are highly variable. Nuclear pre-mRNA introns are often much longer than their surrounding exons. Now, the tRNA introns. Transfer RNA introns that depend upon proteins for removal occur at a specific location within the anticodon loop of unspliced tRNA precursors and are removed by a tRNA splicing endonuclease enzyme. The exons are then linked together by a second protein, the tRNA splicing ligase. Note that self-splicing introns are also sometimes found within the tRNA genes. Dear students, let's now discuss about group 1 and group 2 introns. Group 1 and group 2 introns are found in genes encoding proteins, that is the messenger RNA or mRNA, transfer RNA or tRNA, and ribosomal RNA or rRNA in a very wide range of living organisms. Following transcription into RNA, group 1 and group 2 introns also make extensive internal interactions that allow them to fold into a specific complex three-dimensional architecture. These complex architectures allow some group 1 and group 2 introns to be self-splicing, that is, the intron containing RNA molecule can rearrange its own covalent structures so as to precisely remove the intron and link the exons together in the correct order. In some cases, particular intron binding proteins are involved in splicing, acting in such a way that they assist the intron in folding into the three-dimensional structure that is necessary for self-splicing activity. Group 1 and group 2 introns are distinguished by different sets of internal conserved sequences and folded structures, and by the fact that splicing of RNA molecules containing group 2 introns generates branched introns like those of spliceosomal RNAs, while group 1 introns use a non-encoded guanosine nucleotide, typically GTP, to initiate splicing, adding it on to 5' end of the excised intron. Dear students, let's now discuss the functions of introns. Although introns do not encode protein products, they have very important biological functions. For example, the most important function of introns is that they allow for alternate splicing, making it possible to generate multiple proteins from a single gene, that is polygene. Some introns encode functional RNA molecules through further processing after they are spliced. Introns also play essential roles in gene expression regulation, such as nonsense-mediated decay and mRNA export. Dear students, let's now discuss non-coding DNA and diseases. Many regions of non-coding DNA play a role in the control of gene activity, meaning they help determine when and where certain genes are turned on or off. Other regions of non-coding DNA are important for protein assembly. By altering one of these regions, a variant, also known as mutation, in non-coding DNA can turn on a gene and cause a protein to be produced in the wrong place or at the wrong time. Alternatively, a variant can reduce or eliminate the production of an important protein when it is needed. Not all changes in non-coding DNA have an impact on health, but those that alter the pattern of the critical protein can disrupt normal development or cause a health problem. Variants in non-coding DNA have been linked to several types of cancer and developmental disorders, such as isolated Peary-Robin sequence. This condition is caused by changes in the regions of non-coding DNA that acts as enhancer elements. Enhancers attach proteins that help turn on particular genes. The enhancers altered in isolated Pierre-Robin sequence control the activity of the SOX9 gene. In addition to enhancer elements, variants in non-coding DNA can disrupt other regulatory elements as well. These other elements include promoters, where proteins that turn on genes attach insulators, where proteins that help shape the activity of the gene in different ways attach, and silencers, where proteins that turn off genes attach. While most mutations in the non-coding regions will have no impact, some DNA alterations can play an important role in health and diseases. Some genetic variants in these regions are associated with genetic syndromes, such as myotonic dystrophy type 1, and is caused by expansion of CTG trinucleotide repeats in the non-coding region of the DMPK gene. Variants in non-coding DNA may also be related to more common complex disorders, including heart disease and cancer. Therapies are being developed as research provides more clues as to the relationship between non-coding DNA and diseases. Dear students, it was all about today's lecture. Hope you have understood it well. See you with a new topic next time. Take care. Goodbye.